Cuando quieras.
Ahí.
Perfecto.
Entonces, muy buenos días a todos y a todas las asistentes a este ciclo de conversaciones en estudios ambientales y rurales del Doctorado en Estudios Ambientales y Rurales de la Pontificia Universidad Javeriana. Hoy tenemos nuestra última conferencia del semestre y como invitado tenemos para el cierre en nuestro ciclo de conversaciones en estudios ambientales y rurales al investigador y profesor Mark Krzmarzick, becario actual Fulbright en nuestra Facultad de Estudios Ambientales y Rurales. Mark ha sido invitado por parte del Instituto Javeriano del Agua. Es profesor asociado de Ingeniería Ambiental en la Universidad Estatal de Oklahoma, en Estados Unidos. Hizo su doctorado en la Universidad de Minnesota. Continuó su formación postdoctoral en las universidades de Hawái y la Universidad de Arizona. En la actualidad es docente de la Universidad Estatal de Oklahoma, donde ha generado un programa de investigación en temas asociados a problemas de calidad del agua, incluida la biodegradación, el ciclo del nitrógeno, el tratamiento del agua en la producción de petróleo y gas y la proliferación de algas nocivas en los lagos. La charla que estará ofreciendo el día de hoy el profesor Mark se titula Tools for the Monitoring and Modeling of Harmful Algal Blooms. La charla será dada en inglés. Mark, we are very grateful with you for being here and sharing your knowledge in this cycle of conferences, and welcome to Javeriana at the doctorate program in environmental and rural studies.
Thank you. Yes, and I'm still learning Spanish. Been able to practice a little bit while here, but I still am learning. So presentations in English. This is one of the projects I've worked on for about 4 years. And most often people think of cyanobacteria, which is the same thing here, cyanobacteria. Usually we use harmful algal blooms or HABs to describe the cyanobacteria that produce our toxins. So it's great to be here thanks to the university and Fulbright, Columbia, with some shared funding from ICETEX, which has been a real pleasure to be here and to get to know everyone here in Columbia at this great university. I like to start to recognize my collaborative partners. Dave Lampert is really the person who got me involved into this project. So I owe him a lot of thanks for bringing me into this project because it has been very enjoyable. He is a professor at Illinois Institute of Technology in Chicago, Illinois. James Keith Lofton is a scientist at a governmental agency in the US, the United States Geological Survey, which does a lot of natural sciences, environmental sciences. They're purely a research and science institution, and it's been great to work with them because they offer a different expertise and perspective. Abiram Pengula, who is the guy in the hat in the bottom picture, was the PhD student and postdoc who was the most involved on this project. This was his dissertation work. And then Mawanika Jadobi helped with a lot of the technical aspects as well, who's the guy in the front of the picture here.
Sorry, Mark, a second.
Yes.
Just we would like to share the screen with the audience. So could you give me your USB
Yes,
flash
let's do
drive?
that.
Thank you very much.
And you see a couple of our lakes in these pictures. David Lampert there is being very risky. That's an active bloom right behind him. It was very slimy. He wanted the picture out on the rock. He did not fall in the lake, but I thought perhaps he would. And then this other lake was having pretty good conditions that day in the fall. I'll go ahead and recognize our funding sources. We've gotten a little bit of funding from multiple agencies, including a private company that has helped with some of our lab analysis, Accurate Environmental. All of these people have been very supportive throughout this project. Okay. All right. So we're getting the slides. Oh, that's the wrong one. Sorry, we're getting the slides for this presentation up. Just a second. Yes.
This?
No.
This? Oh.
All right, so surface water quality is a big issue everywhere, including here in Colombia. Lots of contaminants. We have suspended solids, nutrients, mostly nitrogen and phosphorus, but there's also some metals like iron that has a lot to do with water quality and nutrient cycling. Our harmful algae bloom problems, which are occurring more and more often in the United States, in Colombia, and everywhere. Oxygen demand is a big issue for wildlife, especially fish and aquatic creatures. Heavy metals that can be toxic are a concern, where there are mines and other sorts of industrial activities. The big problem with all of these is you really need a lot of data to understand what the problem is, and you need even more data to understand what's causing the problem. So the focus of this talk today is to kind of share some of the data gathering tools that I have been involved with on this project that helps us kind of understand our problems and what's causing them with harmful algae blooms. All right. This problem has been getting worse over time with more intensive agriculture, more fertilizer inputs, more intensity of land use, whether it be grazing or crops or urban activities. With climate change, we have a lot more extreme weather events. In Oklahoma, we have, the state I'm from, we seem to have either a drought or a flood, never in between, always too wet or too dry. That is a drought index on the side there, showing a 9-year time span where we fluctuated pretty strongly between very dry conditions and very wet conditions. Because of that and increased temperatures, we do have more harmful algae blooms. The number of swim advisories from our cyanobacteria has been increasing. And that has caused a whole host of issues, including taste and odor issues if the water is a drinking water source. The toxins, of course, are one of the strongest toxins we know, natural toxins to man. So it's a very difficult problem because we can't use the water source when we're having a bloom. And then the oxygen levels drop, which hurts fish and other wildlife. So what do we have for monitoring approaches? How do we get some of this data? Well, we'll go through 4 technologies we are using and modeling methods. This is some pictures just to get you warmed up. And the idea behind all of this is really a systems-based approach to the problem. What systems can give us the data? What systems can model and predict our problems, these events, so we know about them in advance. With success, these systems could be used for more distant locations where getting physical data may be more difficult. So think about the whole, like, everything comes together here. Our main study areas are here in the United States. So I show you a picture of the whole United States here. And Oklahoma is the state right north of Texas. No one knows where Oklahoma is, but everyone pretty much knows where Texas is. So we're North Texas. And then Kansas, which is the state just north of us. So if we zoom in there, you can see the two lakes. These are both dammed rivers, so they're reservoirs. They're in the same overall watershed. Marion Reservoir on the top there does drain eventually into Grand Lake of the Cherokee, which is the bottom circle. And then that drains eventually into Arkansas and then the Mississippi River. The land uses in this part of the country is a mixture of crops, forest, and grazeland. There's some major cities that you might be able to pick out. This is the city of Oklahoma City. This is the city of Tulsa. Kansas City is up here. Really, the city most impacted in this is the city of Tulsa. They get their drinking water from the same rivers, the same watershed. So they have a high concern right here, this city, of the harmful algae blooms occurring both in Marion and Grand Lake. If we zoom in on that bottom lake first, for our study, we've mostly focused on what's called Horse Creek Cove. It's a little finger of the lake down at the bottom here. And this is its watershed. It has a lot of grazeland with some corn, winter wheat, and soybean crops that are tilled agriculture. And the part of this lake is very shallow, it's nutrient rich. It's where the harmful algae blooms, the cyanobacteria blooms, typically start when this lake is affected. It usually starts first in this cove. So we're going to monitor, we've been monitoring it for nutrient inflows. We've been monitoring it with our unmanned aerial and boat vehicles. And we're doing a watershed model to interpret some of those results. In the farther north reservoir, this is its watershed. It's much more heavily tilled agriculture. There's still a lot of grazeland, but the overall land use is much more farms. This lake has very frequent algae blooms. Every year in the summer, usually in May or June, a harmful algae bloom starts to take hold, and then it kind of goes up and down all summer until sometime late summer or the fall. It finally goes away as temperatures cool and the bloom starts to play out its life cycle. This one has very low water inflows except during heavy rainfall events. So it's a little harder to do a watershed model because it's a little harder to capture the inflows of the rivers, but we still try to do our best modeling on that. And on this lake, we are taking the advantage of this recurring events to try to capture the microbial community at the early stages of an event. So looking at the month, in the 2-week period before we typically see we have a cyanobacteria bloom, what's occurring in the microbial community? When can we detect the first signs that there might be a problem? And how does that relate with our other monitoring approaches? So the first technology is one that has been around for a little bit, and people are heavily using satellite remote sensing. Satellite remote sensing, we're taking advantage of satellites that are circling the Earth and taking measurements of multiple wavelengths of both visible light and infrared light. And with our lake here, this is the Grand Lake, and a nearby lake called Hudson Reservoir. We trained a model, a random forest model, to take that satellite data and correlate it with observations already being taken at all of these little circles at the lake. So we already had a lot of data of the algae concentrations throughout the lake, and so we could use that to train and prove our machine learning model. In these sites, we're measuring chlorophyll, phycocyanin, and turbidity, and that's our ground truth data. That's, that's our physical, we know it's there, and we can use the satellite to see, well, how does the satellite data work for that? The main satellites data we are using is Landsat 8. This is a NASA mission to observe Earth with multispectral imagery. There's observations everywhere, every 16 days at a 30-meter spatial resolution. There's 9 different spectral bands and 2 thermal bands that it's measuring the wavelengths. Some of those are a little bit redundant information, but lots of nice data being produced on a continuous basis. It's accessible data for everywhere on Earth. The Sentinel-2 data is a similar sort of mission from the EU, the European Union. And they also have 12 spectral bands with a little bit of a larger resolution, 100-meter resolution. So between these 2 satellite missions, we get pretty regular readings of the spectral images of our lakes, as long as there's not cloud cover. That's the hardest part of using satellite data. With that spectral data coming in, we tested 3 different algorithms, multiple regression, support vector, and random forest. We validated the models with part of the randomized subset of the information. And then we tested how good the model is with the rest of the data. And using the R-squared value for fit, we could decide what model works the best. And I put Dave Lampert's picture here. He's the expert in this topic. So if you have any difficult questions on the modeling, I might have to refer you to him. The random forest algorithm was our best fit. This uses sort of this randomized tree method. And with part of the data, it will find what model is going to be predictive. The 3 parameters we're focusing on, of course, are the same that we're measuring on the ground: chlorophyll, turbidity, and phycocyanin. And this is the fit of the data. And you see at the bottom there that the random forest regression offers the best R-squared values for predicting the true on-the-ground measurements of chlorophyll A, turbidity, and phycocyanin. So these are the 3 measurements of water quality that we can use to know if there's a harmful algae bloom or not. We've been able to apply these models during a season from February to July 2021. This is the cove, and these are the images of chlorophyll, turbidity, and our phycocyanin predicted from the satellites with our model. This had observational confirmation. As you can see in July there, the bloom was very close to the shore, which was observed by multiple people and by state and agencies. We think that this is working pretty well to use this model to kind of analyze these water quality parameters for cyanobacteria blooms. This has been published since March, so if you want to read more about the use of the development of these models, in the use of our satellite data, you can always start here and then contact either me or Dave Lampert if you have questions. But it's very promising because, again, as long as you don't have cloud cover, you can get a good estimation of the problem. Technology number 2, all right, the cloud data can be frustrating, and the satellites only pass over every like 16 days. We might want other methods to analyze what's going on. So we're using unmanned surface vessels. We've called ours Manuel. That's an acronym, it means something. And we're also using our airborne vessel. We use a drone that can take multispectral imagery of our lakes at a very fine scale, very, very small, 2-centimeter type of resolution. Mostly I'm going to talk about our boat. So our boat there is Manuel. It stands for Mobile, Autonomously Navigable Unmanned Surface Vehicle for Evaluation of Lakes. It weighs about 130 pounds. We store it in the lab. We carry it to the car when we need to use it. It fits into a mid-sized car, as long as you can fold the back seats down, or a truck. It works as long as the waves are less than 3 feet high and the winds are less than 20 kilometers an hour. I'm mixing English and SI units there. 3 feet is about 1 meter. So there's certain conditions we can't use it, but most of the time we can. It can operate for 3.5 hours of battery using a regular car battery. And it cruises at about 1.5 meters per second. We control it by a laptop with open source software written in Python. So we can upload a image of the lake with coordinate systems, GPS coordinate systems, and trace the path it will go on. And then it will start and go on the path. It can go about 1 mile or almost 2 kilometers away before it loses connectivity to the computer. And it can go faster if you need it to. If it has a problem, it's supposed to return home if it loses connectivity, if it wanders too far away, or it loses a GPS signal. The propulsion here is relatively simple. It's a plastic canoe. It's a very common type of small craft, watercraft, that you can use if you want to paddle it yourself. We don't go on it. We hook it up with our equipment and let it go itself. 2 little motors here that are independently controlled, so it can go straight, backwards, left, or right. The electronics on here are not very complicated. This is all equipment that you can buy online. It has a GPS unit, of course, a simple circuit board, and it's in a little waterproof casing in case the water splashes into it. On one end of the boat, we have our sensors. So this is a group of sensors for pH, temperature, depth, dissolved oxygen, turbidity, chlorophyll, and phycocyanide, which are measured with UV light. It also has a ping sonar, which takes depth measurements of water depth. So we know how deep the water is as it's going along its path. I do have a bill of materials if someone wants to buy it themselves. We are actually preparing the manuscripts describing this vehicle with this bill. It does cost a little bit of money, approximately $2,100 US to build and operate this piece of equipment. But then you can set it along a path, like a lawn mower pattern is what we call it. It goes back and forth, or you can do whatever you want. You know, you draw the path it takes, and it interpolates the software. The programming code that was developed by the students will interpolate the data into a gridded pattern or a raster model of all of that water quality data. Here is our Horse Creek Cove in the Grand Lake. Here is the water depth data compared to a manual survey done physically by a sonar on the back of someone's speedboat. They match pretty closely, but we did find some sedimentation problems in our lake that have developed in the last 10 years, which is not surprising. We've had very heavy rainfall events carrying a lot of sediment into the lake. Our water quality, there's our chlorophyll, dissolved oxygen, temperature, and pH in this from that data gathered from the unmanned surface vehicle. So we can have a fine resolution of water quality that's not dependent on satellite data. And it's a little bit more finer resolution to understand what's going on. At our second location, we are still processing a lot of the data, but here is just an example of our drone data from the aerial vehicle, and then our surface vehicle. We have not developed a subsurface vehicle. That's also an idea though, that we've had a surface vehicle that takes chlorophyll measurements. And so we're trying to develop some understanding of the relationships between the aerial, any satellite data we have, and our boat-based data for our drones. All right, third technology, watershed modeling. Watershed modeling is a whole science. People use it all the time. We're using it to understand our lakes, mostly to sort of identify what is the relationships between rainfall patterns, nutrient loading from the rivers, and then our harmful algae blooms. So we have data from precipitation, we have evapotranspiration data for the water that evaporates out. The difference either goes into the ground or it runs off. To understand how much water is coming into the lake, we use a stream monitoring station. This is a do-it-yourself sort of station that looks fancy, but it's not too complicated. We have a conductivity, temperature, and depth sensor. The depth sensor is important because from that, we can correlate that to river volume flow. Like, it's a little bit of math and a little bit of understanding of the cross-section of our stream. We also attach this programmable auto sampler that can take a sample every 2 days is typically what we did. Every 2 days, it grabs a physical sample, refrigerates it, and using power from a solar panel. And then we could collect the samples, bring them to the lab, and do all sorts of other analysis with it. Oops, a little sensitive. All right. The stream monitoring station, these components cost about $240, assuming you can get things like the solar panel secondhand used. The one thing that's difficult is if you get a flood, the whole thing can wash away. So you do want to have the ability to access this in an emergency if you need to. It automatically logs the data, it uploads it online. All of this data from this monitoring station, before we took it down, it's all available online. Anyone can download our data. It also helps having that online because we can see if there's a problem. If all of a sudden it's not getting any data, then an animal chewed through the wires or something, and we can go investigate. All right, so the stream monitoring, yes, physical samples, we already covered that. All right, so we put that into our model. So with precipitation, evaporation, estimate runoff with the graph samples, estimate nutrient loading. From all of that, we can estimate how much nutrients coming into the river or to the lake, and if that has any impact on the harmful algae blooms. We're still developing these correlations and processing this data, but it does seem that there is an impact, but there's a delay. So after a rainfall event, there seems to be about a 1 week delay perhaps before the algae bloom occurs, but we're still working out the quantitative analysis of that on our site. All right. Right now, this is the last calibration data I can find that we have. Our inflow data is overall, it's being under-predicted or over-predicted. So we still need to kind of refine our model to better match our data. Sorry for the messy figures there. Technology 4, all right, and this is my biggest area of expertise. I do know a lot about water quality, but I do know especially the technical details of DNA work. We're trying to introduce DNA monitoring into this as well. Microbial community dynamics, there's plenty of data suggesting that it's important in the life cycle of a cyanobacteria event. So what we really don't know though is what's occurring in those early stages. Usually people wait until there is a bloom, they go out there, they take samples, and then they understand things. But it tells us nothing about what happens leading up to the bloom. You know, were there certain species of other bacteria that grew that are early indicators of a bloom? When can we detect the actual cyanobacteria or not? And how does that relate to our satellite data, the unmanned aerial and surface vehicle data? Does our watershed model have a correlation to the microbial community dynamics, et cetera? We are just starting this analysis. Thus far, we have sequenced the genome of the metagenome of the lake during an event. So we know the genes of the algae at least, and we have lots of safe samples from this last season that we're processing and analyzing. So these are our 4 tools, right? Gathering data. And we're somewhat fortunate in that these lakes are relatively easy to access. They're about a 3-hour drive from the university, which is close enough. We can make a trip in one day, do a lot of work, get back late. But we want to keep going with this data, keep analyzing it, better refine our models, especially with the drone data and the satellite data, which are going to be more useful in more distant lakes that might be harder to access on a regular occurrence. We're trying to learn the significance of internal and external phosphorus loadings. So, you know, what's the impact of the river versus a turnover of the water column? Because there's a lot of stored nutrients on the bottom of the lake that will get stirred up during like heavy wind, which may not correlate with rainfall. And then of course our biomarkers and our DNA method work. All right, so muchas gracias for your attention. And is there any questions?
Okay, thank you very much, Mark. Liliana, our doctoral student, will be managing the questions from the auditorium here and online. So let's start with questions. Questions?
Simple questions are good. If you don't ask questions, I ask you questions.
I have a question, Mark, regarding the idea of finding some organisms that would help you to, if I understood well, to predict a future bloom of algae.
Yes.
So have you had any communities that show you?
We don't know yet.
It's gonna happen or not?
We don't know yet. We have, we're doing a 16S amplicon sequencing right now of those communities. So I don't have that data back. And this first data set from this last year will probably just produce some hypotheses that we then hope to test in future years. So the satellite data, we have data, public data going back 15 years that we can get lots of data, we can correlate it, we can test the model. With the microbial data, we're at 1 year of samples collected. So yes, I hope that I hope by the time that the summer rolls around, we'll have a hypothesis. You know, last year, these communities really came up right before, and last year was a very interesting year at this lake. It took longer for a bloom to occur and it never fully established. So there's a lot of up and down in the bloom. So we'll see, you know, what we can pick out from the noise of the DNA, of the algae bloom, and see if we can come up with some model prediction of what communities might there be. We believe there could be some things related to iron cycling that might be important. And also just the cyanobacteria itself, you know, how early can we detect it? And when we can detect it, can we detect the genes for nutrient uptake or for growth, just general central metabolism genes that really indicate that it's starting to reproduce or not? So we'll see.
Can this model be used in coastal areas like Mexican beaches where there
Yes.
is sargassum proliferation?
Yeah, so we're an inland sort of state, and we focus mostly on the lakes. In marine systems, it should definitely be applicable as well. Now the model may be a little different. Like the satellite model may need to be trained on marine data specifically, and then tested to see if it works. So it might be a slightly different set of spectral images because it's a different sort of species that typically are growing in marine systems. And so I would think that the spectral imagery input would need its own sort of model to estimate if there's impacts or not. So, but I think if you follow the same approach, I think you should be able to do it.
We have a question soon, by Javier Moulton. Any experiences about algorithms or equation used when correlating remote sensor versus field data?
Yeah, so this is the modeling stuff that I am not the main expert on this with this project or any projects, but there is a mismatch of satellite data and in the ground data. The biggest problem with the satellite data, though, is interferences. It's the cloud cover. You have to have a big enough lake section to take a measurement. During the year, there's a lot of greenery. You know, if there's a lot of greenery right around the lake, that can lead to false positive results. Sometimes if there's certain turbidity, that can lead to a trigger of, you know, the cyanobacteria alert system goes off, says there's a bloom occurring based on satellite image, but you go out there and it's not so simple. It's something else is providing those UV wavelengths. So it is, I don't know if that was the question, but I think it's related to the question. But there's these outliers and there's these, yeah, the correlation isn't perfect. And in our random forest model, we still have, you know, 77% R-squared value, which isn't perfect. So it's, you know, one tool to use. But I always caution people on using satellite data is to try to ground truth it when you can, at least, especially if it's a new lake, a new study site.
There's more questions.
If no one has questions, I have a few more. Sorry. When you don't have access to the money to pay for all the combined tools that are complementary,
Yes.
what would be your suggestion about what kind of tool use if you only have the money, a small budget to go with just one?
Absolutely, the first thing is take advantage of the satellite data. I say it's not perfect, but it's still free data. It's out there. It's you can you know, there's program code in with Python that helps even, you know, that's freely accessible code if you know how to code any. I'm not a very good coder. But you can kind of get the data scrap the data for that lake that you're studying and start with that because it's affordable and there's a lot of historical data that you can have. After that, I think that having a at least a system of probes that you can go out physically to ground truth that data. Maybe the whole monitoring station isn't, you can't really put that into the budget, or you're worried that people will mess with it, if it's, you know, somewhere where people are going to mess with it. At least try to get an idea if you're with physical probe data or in physical samples, you know, selected samples with the satellite data to show, okay, yeah, for this lake, the satellite data works pretty well. Or maybe it works pretty well in the wet season or the dry season, but not the wet season. And so you kind of have a little bit more understanding that way.
Thank you.
Yes.
Diana,
Yes.
Sepulveda, a doctoral student, have a question. How efficient was the tools used from the human energy?
For energy, yes. For energy use.
The manual energy invest from people. I think that is the point.
For the manual, the boat.
Ask your question, please, again. No, Diana.
En español.
Do it in Spanish, I will try to translate.
No, en español.
Ok, lo que quiero saber es qué tan eficiente realmente fue la utilización manual que se ejerció para la elaboración de esas herramientas. O sea, en el tiempo, qué tan eficiente realmente puede llegar a ser desde su experiencia pues, a través de la investigación.
Manual o manual?
No, bueno,
Manual.
manual.
How efficient was the in situ sensing for your research using manual? Is that right?
What maybe the contribution to the results?
The manual has been very important for us because it doesn't disturb the water like a manual boat with a motor would. All right, it's much gentler on the water column. And we, in this last year, we have used Manuel a lot. It just runs on a car battery, and for 3 hours, we get it on a mission. We have to keep an eye on it so that it doesn't sink if it hits a log, which has maybe happened. But it's been very, very valuable because it can get us away from the shore into the river and get us a kind of a good detail. Now, what we're hoping is that our aerial drone gives us a good enough quality of data because it has a much broader area to use. But the probe here for chlorophyll and for phycocyanin is, you know, that's ground truth data on measuring the harmful algae bloom concentration. So we trust this data a lot, and it matches any sort of lab analysis of the same. So it's been very efficient, very useful, and very robust. It's we've had a few parts break, but not too bad.
There's no more questions in the booth.
So,
There's no more in the Zoom.
do you have other questions? No? Ah, so you like raising your hand, I was like, yeah.
It's all persons.
Yeah.
You have another question, okay?
Can I access the one where in the webpage that is an article?
Access the model?
Like see the structure and how it was.
Yes. You mean
The research
the
that it
manual,
shows,
the manual or the?
like the whole modeling for this subject.
The modeling.
So the code of the
Yes.
the methodology or
Because I want to study sargassum proliferation, so
Okay.
I was thinking that this could be a good start point.
Yeah, so this paper here does a pretty good job describing our approach to the modeling, and I believe there's some links to the code that we use. So if not, I think I can get you, it's a GitHub link that has the Python code of the modeling, if that's an area that you do.
That would be a great help.
Okay, yes, yes, we're definitely, we share this data very openly in our code because this is a real, no one's gonna make money solving this problem, including us, and we want everyone to have access to anything we've learned.
and maybe you both ended up together working
I might
at
be
Mexico
able-
in the
yeah,
sargassum.
we could-
Yes.
Project.
It's an
Yes,
opportunity to learn from somebody else that have developed a lot of tools like this one.
yeah.
Okay. Okay, thank you very much to everyone for accepting our invitation to come to this last meeting of our Conversaciones en Estudios Ambientales y Rurales. Thank you very much, Mark, for accepting our
Yes,
invitation
thank you for
too.
having me.
Esperamos tener a inicio de nuestro próximo semestre en el 2024 una maravillosa serie de invitados expertos en diferentes áreas de los estudios ambientales y rurales, y en enero estaremos enviando entonces la primera invitación para nuestra primera conversación del año 2024. Creo que ha sido un semestre maravilloso con unos excelentes invitados y esperamos tenerlos nuevamente el próximo año. Que tengan un feliz descanso de fin de año y un próspero año 2024, ahora que estamos ya finalizando nuestras actividades académicas. Buena tarde.